So our first section, so we invited 5 pioneering parents to share their experience and perspectives as the driving force for patient-focused therapy. All of them are my favorite heroes. And they all have very distinct stories behind them, but they have the common feature or characteristics, which is never give up. So our first parent speaker is Yiwei She. Yiwei is a dedicated mother and advocate. Upon her son Leo's diagnosis with arterial neurodevelopmental disorder caused by a TMPO2 gene mutation, she took extraordinary measures to find a treatment. Despite being told that nothing could be done, she founded the TMPO2 Foundations, and through collaboration with biotech companies, they developed a personalized medicine, a personalized antisense oligotherapy for Leo within just a year. Encouraged by Leo's progress, Yiwei expanded her mission to assist other families facing similar challenges. With her background as a machine learning scientist and mathematician, through initiatives like Project Baby Lion, Yiwei aims to revolutionize the diagnosis and treatment for arterial disease in children, striving to make personalized medicine accessible and affordable for all. In a recent initiatives, Yiwei and I are exploring the opportunity to collaborate with hospitals and patient foundations from China through the large patient population in the country and China's investigator initiative trials or IIT program. We are hoping to accelerate clinical practices with more flexibility. Without further ado, Yiwei.
Thank you, Nianwei. Thank you for that introduction. And it's a pleasure and an honor to be here speaking to you all. So I will be telling you about my son Leo's journey, and focusing on the part that everybody I hope is most interested in is how do we get a drug made for a baby. And as Nianwei said, it's a very collaborative effort. So these are just a small number, these are the leads of our teams, but there's a lot of individuals that helped in the effort. Our principal investigator, Dr. Nicole Colfell at Rady Children's. Oh, not this pointer. Okay, thank you. Dr. Colfell, Dr. Dimmick, who was the chief medical officer at Crayon, or former, and then Chris Hart, also the former CEO of Crayon Bio. And there were a lot of collaborators at UCSD as well. So here's Leo when he was born. And I will spare you the diagnostic odyssey because I think everyone here is very familiar with it. But there were signs at birth. And we got Leo sequenced when he was 3 months old, getting the results back when he was 4 months old. And the paper, so he had to be sequenced in a clinical trial. So sequencing and diagnostics is still not widely available or accessible. And this is a problem at a systemic level. But when the clinical trial for the diagnostics came out, there he is. The lone TMPO2 patient, in the study to kind of to find genetic diagnoses for infantile onset epilepsies. And the incidence rate, the prevalence is extremely high. So almost every baby, I think that the incidence rate is up now to 80 to 90%. So many babies with epilepsy have a genetic etiology. And so, that should be a first line tool when you see a seizure in a baby. So that set me off. So people told me there's nothing you can do. And that didn't seem to be true to me. And even if it were, I was not going to let it stop me. So there's also this thing that brain development happens, for young children, for babies and young children. And it's a very, very fast and dynamic process. And so much is going on from even in utero, but also after birth, all of these things are happening concurrently. So we were on a race against time, and our child, luckily for us, he was not terminal. He did not have a neurodegenerative disease. Nevertheless, his quality of life was on the line, and it was the quality of life for the rest of his, possibly 8-9 decades of life. So we wanted to make a drug for him as fast as possible. So there was a crash course in drug development, and this is what the process looks like. So there's the basic science. there is preclinical work, then there's clinical work. And this is, the design of the FDA and the system that we have in nowadays. And then after phase 4, there's all of this commercialization stuff, which actually is extremely important for accessibility and equity, right? So for me, I thought, okay, what is this all for? I don't care. I just want a drug in his brain ASAP. But then, looking backwards, in order for, every child who needs one of these drugs to benefit, we do need to find a way forward for this step, and also, for every single step in this process. So how do you make a drug? Well, we found the cutting edge partners. So Crayon Bio is an AI first, drug discovery and development company. And with my background in machine learning and AI, I was able to read their patent and do the due diligence and have enough confidence in this company that had never made a drug before. But their patent looked, solid. And so we signed a contract with them and we started working together. And we also, started the whole system of model organoids. So this is the other half of the drug development process, is trying to find, a model of the disease in something other than patient, so that you are not taking the risk with the patient. And so we didn't make Leo yeast, we made Leo worms, we made Leo flies. Actually, Leo flies were already made, but we had to make, more faithful Leo flies. We made Leo mice, and we turned down a proposal for Leo monkeys. They were not only extraordinarily expensive, but then you have to, once you have the monkeys, you have to take medical care of the monkeys, which is also actually very, difficult. And the most promising, and then what ultimately succeeded, were Leo brain organoids. And so we were very, very thankful to the Motri lab and its collaborators. But these organoids, they come from iPSC, so they come from stem cells. And so the first step is to make the stem cells, which iXCells is a great resource for. And on the other side, concurrently, we were making the leads and we were qualifying the leads, and we were going through, this is a standard workflow for ASOs. There's a different workflow for different modalities, but the shape generally looks the same. You start with some leads, this number might be millions for small molecules, or smaller for gene therapies, but then you select down, you try to design rationally when you can and engineer when you can, and apply AI when you can. And increasingly, AI is more and more applicable and effective at each stage individually. And also for ASOs, the company that we worked with, Crayon Bio, they could, out of 100 leads, they could select over 80 of them were safe in kidney and liver tests. So that is a very strong bias towards safety, even at the first step of the screening process. Our drug necessitated allele selectivity. That is a technical point I won't go too deep into, but we found a drug that could hit the target with a very beautiful, dose response curve. So this is what it looked like. This is the Gantt chart of everything running in parallel. And this was, we wanted Leo to have the best drug, as fast as possible. And there's a lot of failure. So there's a lot of hedged risk. So I guess, it's not, I don't want to say failure. I want to say, we mitigated a lot of risk. And so if things happened differently, we would have still gotten to, our end result within about the same time frame. And that is what a significant amount of resources were expended on. So ultimately, the disease model, actually, so yeah, so, everything other than the top two is disease models. And all of these disease models, they all failed for different reasons, except for the the iPSC-derived organoids. So we barely succeeded in some way. So this could have also failed, and then we would have had no disease model. And then the question would have, or the decision would have been, do we go into the human? Do we go into the clinic with no wet lab backup data and only on the back of some bioinformatic data? And that is, that is a harder question to answer than what we had to ultimately answer. And so here is, thanks to the Motri lab, this is the on the right-hand side, you can see that these are the Leo brain organoids. The vertical axis is the diameter, of the brain organoids, and we see that his microcephaly phenotype is recapitulated in these brain organoids. And this is actually, these brain organoids are a very faithful representation of late fetal brain development. And so in all cases of microcephaly, which occurs with an incidence of about 1 in 10,000, this is an excellent, disease model, a place to try to find a disease model. And then our rescue data also came back to be very, promising. So the red line is the control. So, this is what happens to the brain organoids if you don't apply anything. And then we have 3 different ASOs at different concentrations. And we have 2 time points. And we see that ASO B in particular has very good performance because we are not only clearing the 50th percentile, there's a big jump in the 50th percentile, but there's also a big jump in the 25th percentile. And I think actually we got our dosing a little bit wrong. These 5 to 10 micromolar doses are a bit too high. Sorry, I'm jumping into the technical. I'm a scientist by training. But this rescue data was extremely promising and encouraging for us. And it came in January of 2023. So this was after we had the leads. Yeah. And ASOC, it was a control ASO. So this is just a scramble and a non, it's not supposed to do anything. And we see indeed that, it doesn't do anything. And in fact, it's a bit harmful at the highest dose. Okay. So with that, onto the clinic. Well, so between January, this is in January of 2023, and July of 2023, there is the 6 months of applying to the FDA. So, that is one of the problems in this space is that we have safety data in non-GLP trials. We have efficacy data in patient-derived cell models, but it took 6 months for us to get into the clinic. And something about that risk-benefit, outcome doesn't sit right with me because Leo was having just, sometimes up to a dozen seizures a day. And often he would stop breathing during these seizures. So there's a conversation to be had with FDA about how they regulate these therapies. And so here is our clinical protocol. We doubled every month during the escalation phase, again, for safety. And then after that, we did every 3 months at the maintenance dose. This is again a science slide, but it's trying to find the pharmacology. So trying to figure out, are we hitting the target in the CSF, in the brain? And this is a assay that measures RNA in the CSF. So there's actually a tiny amount of RNA in the CSF, and it turns out there's maybe five molecules of the TMPO2 transcript. This was 200 milliliters of CSF. So there's a very minute amount, and we tried to measure it, and it's incredibly noisy. So, at one point there was 100% of it was the mutant RNA, and at a different point, only 25. So only like 1 or 20%, 1 in 5 of the transcripts. So this is, we tried our best to measure it, but it's very hard. And, this is one, indication of the challenge of trying to do these trials scientifically. Next slide. There. Okay. So seizure data also came out to be very promising. We saw the decrease in seizures, especially these dark, severe grand mal seizures right away. The second thing we noticed was that the seizures would recur about a month in to the 3-month maintenance cycle. And I think the issue there again is kind of it's both the FDA and our lack of pharmacology study before we went into the patient. One thing that helps FDA applications is if you hew to precedent, then they will approve you. One thing that does not help you is if you're proposing something new, and then you might get bogged down into a conversation. So we hewed to precedent, and we proposed a protocol that was, that had been submitted and approved many times. And so, this between these 2 doses, this is when we tried to have a conversation with FDA about increasing the frequency, which they did approve. So thank you, and kudos to them. So we are now dosing every 6 weeks, and we have been seeing increased seizure control as well as developmental progress. I should skip this. And yeah, so the last piece is, the thing that we as Leo's parents, thank you, as his parents, wanted most out of the clinical trial was not seizure control. It's it's neurodevelopment. And so this is one of maybe the other half, or maybe the main challenge is how do you measure, childhood development scientifically at an N of 1, scale. And so some of the things we did, well, so these are the successes, right? So we got a new milestone, which is he went from not having head control to being able to sustain a tripod sit, which is, a significant improvement. So he can't get himself into the sitting position, but if we put him there, he can balance himself. There's less spasticity. This is both an objective and a subjective measure. So with AI and newer technologies, we can, have objective metrics for spasticity. He has new social emotional developments where he will laugh and cry with his sister. So the crying was very easy to notice. Before, I had one child crying at a time, and then all of a sudden, whenever I had one child crying, within 5 seconds, I had a second child crying, and it was very fun actually to have this experience because I knew what it meant. Strabismus, a lot of these children with neurodevelopmental diseases, there is some manifestation in the eyes. And this is another opportunity for an objective metric. And again, the developments in AI, especially these multimodal models, can make, a clinical measurement of this kind of a symptom, objective, cheap, and easy. You just have to tell the model what you want, and it will measure it for you. This is really hard, but we noticed, a big jump in responsiveness and presence. He went from a baby who sometimes, we would ask, like, is he in there? Is someone in there? And I know a lot of parents have this experience to a baby. We think, yeah, there's definitely someone there, and there's something he wants, and, and all these things. And in terms of there's something he wants, he will attempt to reach for toys. He will interact with toys. He will interact with sounds. He will notice cause and effect when he makes a sound. And, we are really excited about the new sounds that he's making himself. And since the increase in dosing frequency this past summer, we've noticed there's less regressions between doses, and some new sounds that are consistent. So I think, in terms of development data, in terms of clinical data for childhood development, I don't have a complete answer, but the tools are becoming available, right? So there are tools for getting biomarkers for EEGs, for blood sampling and other, biomarkers that you can take at higher frequencies. So there are, there are real, blood droplet tests, which you can send to like, say, the Snyder lab, and you can measure hundreds of different biomarkers in the blood. And there's now, dry scalp EEGs where you can measure, at a high frequency, at a high resolution, so that you can get objective data from a single patient, and it can power a whole trial from just a single patient. The data from a single patient, let me say it this way, the data from a single patient can power an entire trial if you take the measurements frequently enough. Again, acknowledging the team and the takeaways. If nothing else, I hope you take away from this that the neurological symptoms are almost always genetic. So doctors are somehow behind the curve on this. The parents know this, and we should somehow inform as many doctors as we can. AI is becoming, ubiquitous. and it's really good for rare diseases because it's incredibly deflationary as a tool, and the economics of rare disease has always been the biggest barrier to therapies. And so AI at almost every single stage of the pipeline of drug development, from the literature review to the discovery to the development to the clinical trial to just clinical care and practice, is it's entering everyday practice. And I hope that rare disease parents, advocates, researchers can leverage the productivity and scientific gains that are now available, and find the most creative ways to apply it to our field and our children. Yeah, our partnerships were critical. Our partners, all of the people who were part of Team Leo, we couldn't have done this without any one of them. And the standard of care needs to be better for rare disease patients. Standard of care does not cut it. And again, this is where AI can outshine human doctors and human knowledge by so many orders of magnitude that I think it's enough. The change in quality is enough to overcome the inertia and the other barriers towards adoption. Being this end-to-end process, so we know we have all of Leo's patient information thanks to HIPAA. We have all of the costs of his care, and this was the cost of standard of care before the trial, right? So this is what was built to insurance, and this is not even including any of, say, the lost work time for the parents. This is not including the reduction in quality of life. Leo, had he been a healthy baby or treated earlier, he could have been a productive member of society. That is an economic cost. There's all this therapy that the state provides him. There's caretaking for probably the rest of his life. And so these are, when you add this up, it comes out to millions, maybe $10 million. I don't know, but it's a number that is not so much less, maybe the same as the cost of the drug development program. And so, when we as a society and our representatives and our policymakers and all of these people think about this, maybe the economics of creating drugs one at a time, one patient by patient, they're not so far off from break even. And if that's true, then why aren't we doing it that way instead of dumping a lot of this money into anti-epileptics and all of these these specialist referrals that ultimately really did not have any impact on his quality of life. And last, so I am a mathematician and a machine learning engineer. So this first principles concept, trying to do things from first principles, this is kind of a core part of who I am. And that is how I went into this project when he got diagnosed. So all of these are kind of, if you were a scientist and you wanted to solve a problem, you might not start with, well, this is how things have always been. You might start with, what are the constraints? And are there any constraints? Are there any laws of nature that prevent Leo from being dosed in 2 weeks? No, there aren't, right? What are the constraints? There are regulatory constraints. There are operational constraints. There are economic constraints. But these constraints can be overcome in ways that laws of nature cannot be overcome. These are human constraints. And we as fellow humans can work together to remove these constraints so that in the future, one day, maybe in the near future, these children can be treated within weeks of being diagnosed by a drug that is safe and efficacious, and the whole life can be recovered and rescued. And that is a vision that I deeply believe in and would like to work and am working towards. And technology and the advancement of science will make this future possible. I think that's it. Yes, that's it. Thank you so much for your time.
Thank you, Yiwei. I saw there's some questions pop up from remote, but we do have a Q&A section after each session. So there will be 20 minutes on Q&A after that. So we were gonna hold on that. So really appreciate Yiwei's experience, sharing her experience. So I also appreciate your squeeze your time. I know she's gonna go rush to Lady Hospitals for the next dose for Leo. So, and the whole event is pretty much scheduled on this day because of you. And thank you. That means a lot for us. So our next speaker, Michelle Jeanette. She's also one of my favorites, really favorites, the foundation leaders. She is the founder and president of Cure KCNH1 Foundation. Michelle is a passionate advocate with a crystal clear mission. Her goal is to create a meaningful impact by raising awareness about KCNH1 genetic disorders and positively influencing the lives of children and families. And she is unwavering in her commitment to drive research, constantly seeking improved treatment options and holding on to the belief that a cure is within reach. With relentless efforts, Michelle has established a collaborative network of partners, including Lauren, iXCells, and Parora, and many other organizations for developing therapies for KCNH1 kids through different roadmaps. Without further ado, let's welcome Michelle.
Okay, perfect. All right. Hi, everyone. I hope I can get through this without coughing. I had pneumonia over Christmas and my lungs are still not anywhere close to recovered. So I want to share a bit about our family story. These are my 4 beautiful boys. I had always wanted a big family. You know, I dreamed about all the normal things like camping and hiking and sports games and movie nights and beach days and family dinners. And we were lucky to have 3 healthy boys. And then Tristan came along in November of 2019. And I took one look at him and I thought something looked a little off about his eyes, but none of the doctors or nurses mentioned anything. So I didn't think anything more about it until he started missing developmental milestones at about 4 months old. We got him into PT and OT, and we got referrals for genetics and neurology. And the first rounds of genetic testing came back negative, and he had clean EEG. So we figured, you know, it must be some mild thing, minor thing, and with lots of work, he can catch up. And then he was about 15 months old when we got his genetic diagnosis, and it turned our lives upside down. He was not that far behind at the time, and so we had no idea how severe it would be and how it would essentially change every minute of our family's life forever. And then about a month after he got his diagnosis, he started having his first seizures, and that changed everything again. We are lucky that Tristan is relatively stable compared to a lot of rare disease kids, but we have still had numerous ER visits and ambulance rides and a couple of hospital stays and canceled trips and really hellish medication side effects that he's had to go through. And in addition to all that, it's all the everyday other effects of rare disease on our family's life that are really, really hard for us and all the other rare disease families. I picked this picture of our kids at the beach because I think that's just about the one time in Tristan's life that we have all been able to enjoy an activity together as a family. The stars aligned and he was actually enjoying the water and the temperature was okay and he actually had fun, which was amazing. But Normally, he basically can't participate in any normal activities, or he doesn't tolerate them. And so it means that we always have to divide and conquer. Somebody has to stay home with Tristan, or we have to line up a sitter for him. And it breaks our hearts that he can't participate more in life. It's also really devastating that he spends 20 hours a week in therapies and he makes essentially no progress. I don't think he's learned a new skill in almost 2 years. He spends holidays zoned out while our other kids are, tearing open their presents. And one of the most stressful and heartbreaking aspects is that he just constantly cries and screams, and no matter how hard we try, we can't figure out, we don't know what's bothering him. He's completely nonverbal, so we don't know what's bothering him or how to help. And it's extremely heartbreaking to see him and stressful on all of us and our other kids. So as soon as Tristan was diagnosed, I decided I was going to do everything possible to create a better future for him and for our family. So I started reaching out to researchers all over the world. We started the process to create a foundation. We started fundraising. I started doing a million things that I never thought in a million years I would be doing, like learning molecular biology and electrophysiology and neuroscience and grant writing and event planning and website design and meeting with local leaders and advocating at state and national levels, public speaking. And we've really come a long way. I want to back up a little bit and give some context about our disease. KCNH1 related disorders are an ultra rare developmental and epileptic encephalopathy caused by mutations in the KCNH1 gene, which encodes a potassium channel primarily expressed in the central nervous system. We have about 100 cases diagnosed globally, and it's very similar to a lot of the other DEEs, characterized by intractable epilepsy, profound intellectual disability, severe movement and speech disorders, autism, GI issues, feeding difficulties, sleep disturbance, and then we have a couple kind of unique features to KCNH1, which are gum overgrowth, which requires repeated surgeries to address, and smaller missing finger and toenails. Most of the mutations studied have been shown to be de novo variants that cause a gain of function, so the channels open too easily and stay open too long. And KCNH1 also has a connection with cancer. 70% the gene is overexpressed in 70% of tumorous cancers. And so it's possible that any of our treatments we develop that either downregulate the gene or inhibit the channel could potentially help cancer patients. Our foundation is really laser focused on getting translational treatments to the clinic. But of course, to do that, we also need to do all these other things in parallel. We need to generate models. We need to collect patient data and establish the natural history of this ultra rare disease. And we need to organize and engage the patient and professional community, and of course, raise money. Sorry, it's got a lag. I can't tell if it's gone or not. Oh, okay. So when we started our foundation 3 years ago, no one was actively working on translational treatments for KCNH1. We've made huge strides, and we now have multiple modalities in development. We have a repurposed drug that Tristan and a few other patients have tried with mixed results, some having great benefit, some none, some adverse events, not serious, but and side effects. Tristan has experienced some benefit, but nothing life-changing. We funded a repurposed drug screen and identified a couple promising hits that now need to be validated. We designed, manufactured, and screened a batch of general knockdown antisense oligonucleotides, but what we're most hopeful for Tristan is that he was accepted into the Enlorum program for the development of an allele-specific ASO, and I hope we might be hearing a bit about that later today from Nadina. We have academic and industry partners working on novel small molecules that are potent and selective channel inhibitors. And one thing that I'm really excited about for the long term that theoretically is the most curative option is base editing. And our foundation just received a $100,000 grant to fund a pilot base editing project. And the preliminary data is really exciting. We have a couple highly efficient editors. Okay, our disease models. Basically from the beginning, we started working on disease models. And we now have simple cell lines, we have iPSCs, we have neural networks, we have cortical organoids, and we have a couple mouse models. But you know, everything takes a while, and with delays and various issues, none of our disease models are fully completed and characterized other than the simple cell lines. And so we haven't really gotten to that place where we can put all of these exciting treatment candidates into our disease models and test them. The theme of today is collaboration, and we have been so lucky with our foundation to have numerous academic and industry partners who have been super collaborative and wonderful. But I know a lot of my friends in the rare disease space have not always been so lucky. And so our priorities as patients, of course, are urgency and involvements. We need better treatment options for our kids now. They don't need to be perfect. We know they won't be. We know there will be risks and that there could be adverse events, but we are willing to take those risks because our kids are suffering. And we ourselves, personally with Tristan and countless friends in the KCNH1 community and other communities, have encountered so many barriers in just trying to try repurposed drugs. So for example, Tristan, the drug that he's on is on label for pediatric populations for bedwetting. You can hardly imagine a more benign indication. And yet we jumped through hoops for a year. We did all kinds of in vitro tests, medical tests, developmental tests, consulting everyone and their brother, all of whom were supportive. And then the neurologist that we had specifically been working with, because she's in the translational space, got cold feet, didn't want to prescribe it, and it was devastating. So, not all families are going to want to take risks, and that's fine. But for those of us that do, we need advocates and allies who are going to be supportive of that. And then, of course, we all know that it's very important to involve the patient communities in clinical trial design. It leads to the selection of better endpoints and outcomes and more successful clinical trials. Some of the things that have been really wonderful with all of our partners that we've worked with is communication. When the research you're working on literally determines your kid's future, just being kept in the loop, even if there's no news, makes all the difference. And then focus on the big picture, share everything possible. We've had partners who have been so generous with their time and their info and their assets. So, don't let competition get in the way of progress. And then take the time to invest in the patient community and get involved. Show up at our research roundtables and our events and even our fundraisers. This builds trust with the community and it motivates everyone. I've heard so many times how meaningful it is to researchers to get out and see the real world impact of what they're working on. And likewise for the patients, it makes all the difference to meet the people who are working on their kids' disease and build those connections. So yes, it takes time, we're all busy, but I promise you it will be worthwhile. I think we all know many of the benefits of collaboration, access to the patient community. We've hooked up our researchers with many patient samples, data collection, clinical trial recruitment. I do not enjoy being a professional nag and hounding people down for survey completion or lining up a blood draw, but I will do it and I can help take that burden off of you and probably be more successful at it because I have those relationships. Of course, working together, meeting regularly, breaks down data silos, reduces duplication of efforts, and avoids waste of our minimal resources. We've been really successful in working with our scientific advisors on funding applications. So we've submitted letters of support and patient cell lines that have helped some of our academic collaborators win multi-million-dollar awards. And in turn, our scientific advisors have helped us, edit and review our grant applications, and very much helped us win the really large CZI grant this past fall, as well as the $100,000 one for the base editing project that then we, grant back to a lot of these researchers that we're working with. So it's a really mutually beneficial relationship. And then, of course, again, meeting regularly really helps everyone optimize their research protocols and, get questions answered quickly or come up with new insights that you just wouldn't, likely send an email. But if you're all connecting on a regular basis, it can really help accelerate things.
Hi, my name is Hazel, and I have KCNA4.
Can you say hi? Say hello. Hi.
Hey.
Hi. We call him Daddy's tail because he follows him around everywhere. And he's trying to catch up with me because he'd rather be holding my hand.
Stella may not be able to walk independently, but she knows how to use her new walker to get what she wants. She even managed to make off with everyone else's coats at school one day.
She smiles in the face of adversity.
Tristan is the sweetest, most loving little person. He's always toddling over to lay his head in our laps or grab us as we're walking by.
Casey and H1 affects everything about Joseph. It makes him who he is. And it affects, obviously, everything about our lives as well. Our lives as a family are maybe not what we thought they would be when we had our little boy all those years ago.
Mais c'est ça, malgré son âge de 16 ans, c'est que elle a quand même elle a un âge mental de 8-10 mois, ce qui est très difficile. Elle fait rien toute seule, elle dépend de nous dans toutes les sphères de sa vie. On l'appelle notre bébé géant d'amour. Fait que c'est sûr, s'il y avait un médicament qui pourrait alléger, faciliter plus son quotidien, puis notre quotidien, mais c'est ça, ça serait, j'aimerais vraiment qu'elle ait plus conscience de ce qui l'entoure. Qu'elle puisse nous dire je t'aime. Que j'ai l'impression qu'elle nous reconnaît, sa famille. J'ai pas l'impression qu'elle nous reconnaît. Puis ça me fait de la peine des fois. Tu sais, mettons qu'on va la chercher à l'école, on arrive à l'école, c'est comme si on était des purs étrangers, là. Elle semble même pas nous reconnaître. Ça, je trouve ça difficile. Je trouve ça vraiment difficile.
Onun gelişim basamaklarını tamamlayamadığından itibaren hem özel eğitim hem fizyoterapi aldırdık, ancak ilerlediğimiz yol çok uzun bir yol olmadı. Bu yüzden de dünyada yapılan bilimsel çalışmalardan da umudumuz yüksek. Belki geliştirilecek bir ilaç ya da genetik bir tedavi Melih'in yürümesine, konuşmasına ya da farklı bir şekilde iletişim kurmasına.
My brain sends a message to my mouth that my mouth doesn't get.
I would say that the most difficult part of having a child with this disability is the unknown. Bentley could have just, a wonderful start to his day like he did today. A little bit tired because he decided that he was going to stay up all night, to, having a full-blown grand mal seizure this morning, which is very scary, very traumatic.
The hardest part
which is Rafi's seizures. They're extremely scary. My husband and I feel often very helpless because there are no triggers, many times no warning signs, and even at 8 years old, they're really uncontrolled.
Mi mayor esperanza, me diga lo que quiere, lo que siente, lo que le gusta.
So if we could eliminate seizures altogether, that would be a dream. Our hopes for the future and with future treatments are for Bentley to have cognitive growth. To us, it's more important to have that cognitive ability than physical abilities. So if treatments could help with that, that would be a dream come true.
Love for Arisa to be able to understand us and talk to us, but in the end, it doesn't change the fact that she is our sunshine.
So thank you all for learning a bit about the KCNH1 community, and of course, this is representative of all the communities we serve. I just appreciate everyone being here and all of the work that we're doing together to improve the futures of the rare disease community.
Thank you, Michelle. Every time we're here, Michelle's story is very, very touching. We've been working with her for many years, and her story has been inspiring, not only my company and also my families. And today I have two of my children here today, Laura and Andy. So sometimes they have the school assignment to ask their parents, what do you like about your job, right? So I tell them, right, it is the mission, and there's so many things in front of us, right? So, and we're lucky that we're in an era that there's so many scientific breakthrough in front of us: gene therapy, cell therapy, iPSC, and CRISPR. So many things that we can potentially bring those, which is, personalized medicine, which is unbelievable a couple of years ago, but now it's becoming, we already bring this treatment to life. So without further delay, I'd like to introduce the next speaker. His name is Piotr Kosla. He's dialing from Poland. So he's a proud father of 3.5 years old twin girls, Lena and Zuzia. Piotr comes from Poland, and where he currently lives with his family. His professional background is finance and international management. He graduated from Warsaw School of Economics and National University of Singapore. Before joining Biogen, he spent several years in finance and business development in several big firms. 3 years ago, Piotr's daughter Lena was diagnosed with the ultra-rare genetic disorder, PACS2 syndrome, PAX2 syndrome. He and his wife founded the PAX2 Research Foundation, driving treatment discovery for about 150 people affected by this disorder. In 2022, he reached out to us for iPSC generation for his daughter without knowing too much background of this disease. And ever since then, I've been following his journey in developing the treatment for children with PAX2 condition, and was super impressed by what he has achieved with only 3 years. And today we are going to share he's going to share his journey with us. Peter, can you dial in?
Yep. Hello, everyone. First of all, can you hear me and can you see me? That's the most important.
We can hear you. We're working on a slideshow.
Okay, perfect. So let me first, okay, I'm trying to share my screen. Wait a sec.
Yes.
So as long as you share the screen, I cannot share my screen. So you have to stop sharing the screen.
Yes, hi, Piotr. I just stopped sharing.
Almost there.
Perfect. Okay. Okay.
Let's see how it goes.
Perfect. We can see that loud and clear.
Okay, that's great. Great, that's great. Thank you. So first of all, welcome everyone. Good morning to everyone. I guess it's around exactly 9 a.m. On my side, it's 6 p.m. So I'm dialing in, as it was mentioned, all the way from Poland, which is 1,000 miles away. But it doesn't stop us from collaborating together with iXCells and with many other parties, which I'm going to mention during my presentation. But before I get there, I wanted to say a big thank you to Michelle for having a great and very emotional presentation. And she always inspired me as well in my job around the PAX2 when I see what she's achieving and what steps she's making with KCNH1. So thank you, Michelle, for that. And I hope at some point we'll get to meet each other in person. Because for those who don't know, we've met like 2 years ago. And every now and then we update each other, how we're doing. And I'm always inspired by amount of motivation that Michelle is having to push the children development further for all the kids with the KCNH1. So thank you for that. Moving to our case. So today I want to talk for, let's say, 15-20 minutes about the journey we did in the last 3 years when we're starting completely, and I mean it by saying completely from scratch, when there was like no interest around the PAX2 syndrome at all back in 2020-22, and where we are today after 2.5 years, to be very specific, from the moment we decided to do something about it. So as it was mentioned, me together with my wife, so my name is Piotr Kośla, and with my lovely wife on the left-hand side, Małgorzata, Gosia Kośla, we do have some background, which in fact helped us to, let's say, start a bit faster than some of the other parents that decide to take care of developing the drug for their diseases, because I have some medical background, as well as my wife. However, both of us, we finished business schools. We didn't study medicine. We didn't study biotechnology and any of that. So we had to learn very, very fast. But something I'm gonna link during the presentation. I'm going to link the fact that the fact that we finished the business school helped us a little bit at the very beginning to, let's say, to manage a project. Because at the end of the day, I guess every drug development is a project. That it also requires specific competences to push it further. And on the right-hand side, you can see our reason. So Lena, that today she's 3.5 years old, but she has twin sister, which is very important here, also from the research perspective. They are fraternal, as you can see also from the picture. But in fact, it allowed us to move quite fast with the cell painting research, which I'm going to talk about later on, and how Zuzia cells were beneficial in advancing the children development for PAX2. But starting from the very beginning, so basically our story. So before December 2021, we're a normal family with two kids, with twins, which were back then 3 months old, and we didn't see it coming that at some point Lena will have the first epilepsy, which very, very quickly moved into the status epilepticus. Because of the fact that they were twins, some of like Lena, she had some issues with the tension in the body, together with Zuzia, but it was always explained by the fact that they are twins. So as simple as that. So, and apart from that, everything was fine. So if you look at Lena when she was 1 month old, 2 months old, 3 months old, you wouldn't tell that she might be having some devastating disease in front of her. But in December 2021, during the holiday time, she experienced her first epilepsy, which followed by 30 plus in 48 hours when she was in the status epilepticus in the hospital. And then quite quickly, we've managed to diagnose her thanks to whole exome sequencing in February 2022 during Valentine's Day. So it was exactly 3 years ago, as we have 18th of February today. And we quickly learned that she's going to face a very rough and unpleasant future because of the clinical phenotype that was described in the first publication from Heather Olson. And when you read the publication, you see that she will be facing intellectual disability, autism spectrum, psychomotor delay, cerebral dysgenesis, and so on. So very similar to other DEEs as well, and also to what Michelle was speaking about like minutes ago. And back then, Lena was second in Poland, and around 40 case diagnosed globally. So as you can imagine, the interest around the disease was very limited. Back then, when we decided to start the foundation in May 2022, there was 0 scientific publications on the disease, apart from the fact that there were some case reports, and only one scientist, one professor interested in the PAX2 syndrome, and only because he was studying healthy gene. So at some point, he also decided to spend some time understanding the syndrome itself, and that was it. So that was our starting point. So really from scratch. And then I will tell you, how we, what kind of, what kind of progress we made in May 2023, May 2024, and where we are today in our journey. And at the very beginning, that's all we knew about PAX2. So we knew what is the healthy gene role, what is the protein structure, and as you can see, the PAX2 is located in a highly unstructured region, and that there were like cases of mutations in E21IK and E11K, which were leading to the disease. And the last thing was that the PAX2 gene expression was highly expressed in brain and skeletal muscles. And regarding brain, it was mainly oligodendrocytes, and that was it. So this is what we started from, and also from case reports, which were first described by Heather from Boston Children's Hospital, explaining how the clinical phenotype looks like. But today, before I move to the scientific part, today we are connected to more than 150 patients worldwide. And as you can see, not only we are connected to the patients, but we are also connected to the physicians, because building on what Michelle was saying about the natural history study, we are also preparing for that, because we are trying to tackle everything at the same time. So not only focus on the research itself, but also looking from the clinical perspective, what will be needed in case treatment is ready. However, what we see is that the same mutation variant, because 95% of the cases of PAX2 are kids with E21IK mutation, really like 95 or even 98% of the cases. However, phenotype differs a lot. So we have different onset of epilepsy, we have different spectrum of intellectual disability from mild to severe, or even we start to see patients with borderline or even almost normal intellectual development. You can have autism spectrum disorder. Most of the kids, they have it. And the same with verbality and non-verbality. More than 3/4 of the kids are non-verbal. So, but the striking thing is that despite the same variant, you have a really very, very phenotype of the disease. And we also try to understand why is that happening. And in May 2022, when we decided to start the whole journey with the PAX2, this is where we were in terms of the reagents. So it was quite lucky that we found out very quickly that very close to Poland, in Czech Republic, today called Czechia, there was actually non-humanized mouse model which was frozen, and we found about it by coincidence in a way, and that was our kind of situation that hey, so we kind of have potentially non-humanized mouse model with the PAX2, which is frozen. Let's do something about it, and then this is where we are today. So. Down the line, when we decided to start the whole journey, we got into collaboration with couple of different places all around the world, including iXCells, because as Nyanwei mentioned, in fact, the fact that we are here in Poland and it didn't stop us from sending the blood all the way via FedEx from Warsaw to San Diego. And in fact, the first iPSC line was generated with iXCells, which was then in person delivered by Nyanwei to Northern Foundation, which I'm going to talk about in a second. But today we are basically we have I mean maybe not all, but most important reagents to work on, meaning fibroblasts, APC lines, neuronal cells, both humanized and non-humanized mouse model. mouse model, and with some of them, like for example, you can see here with the non-humanized mouse model, we actually did some initial phenotyping as well. So we are kind of, try to move as fast as possible. But what is very crucial is that from the very beginning, we have this approach of doing everything at the same time. So we focus on basic research, translational research, clinics, all at the same time. And from our perspective, it's all about the cooperation. It's all about reducing the bottlenecks from administrative perspective, and also maximize the investment. So we are looking at every dollar from every side. And what we also do, we try to monitor all the work on the PAX2 syndrome across the globe. So in case there's something new happening around the PAX2, very often researchers, scientists, they approach us first, and we straight away we update them what is happening in all the places in the whole world, so that we don't duplicate the job, and we allow this flow of the information between everyone involved in our journey. So as you can see here on the slide, we are focusing on the in vitro, in vivo aspect. From the translational research, we are super lucky to be collaborating with the NLOREM, because Lena is part of NLOREM family since late 2022, and that's the most promising approach in PAX2 syndrome. Silencing the mutated gene, or the whole gene as such. But we are also trying to approach the problem through drug repurposing, and I'm going to talk about it in a second. But we are also looking at more long-term perspective. I'm talking, 5+ years from now, regarding RNA editing, genome editing in mice. And last but not least, as I mentioned, we are focusing on collaborating intercontinentally on the natural history study, because with such limited patient pool, sometimes it's sometimes it's impossible to do the proper natural history study within one country because of the number of the patients. Because obviously doing the natural history study with 2, 4, 5, 10 patients is not enough. So that's why from the very beginning we work intercontinentally, as you can see here, for example, between Boston Children's and our medical university here in Warsaw. And we are part of many different initiatives like EpiCare, which is epilepsy care in Europe, or Simon Searchlight. Today, we do have some progress around PAX2 on many fronts. So first of all, we have a couple of compounds as a potential drug repurposing candidates based on our cell painting study research with Charles River. As I mentioned already, but I'm gonna mention it once again. Lena is part of NLOREM family, so we are part of this active program for PAX2 syndrome. Hand in hand with many different diseases, including KCNH1, as Michelle mentioned. We do have the mouse model ready for the preclinical examination, but we are also focusing on things like publications. We do know it's important also for the researchers to publish about their job, and we try to do anything we can to support it. And we are also focusing at some point to move from crowdfunding and using our private funds, because something that I didn't mention is that 95% of things we're doing is our private money, either our private money or crowdfunded through friends and friends of friends and so on. However, we are doing also some progress around the grants. So we got 2 grants awarded for basic research in 2024 from NIH. So we're actually helping or closely collaborating with researchers that actually got them from Pittsburgh and from Chicago. However, we are not stopping here. We are in process of submitting or awaiting decisions for different grants all around the world, including US and Europe. So kind of to summarize, before I move to the video, summarize the situation right now. So from May 2022, with one interest among scientists and one project of the PAX2 syndrome, today we have more than 50 scientists which are somehow involved in the disease, and more than 10 active projects happening all across the world. Which I wanted to kind of highlight here is that it doesn't matter that we started from 2 patients in Poland and 40 patients in the whole world. I tend to say that today more scientists are involved in the project than number of patients diagnosed 3 years ago, which for me is amazing to be able to say that. And also to kind of to send the message out there is that, you know, at the end of the day, science doesn't choose, and science doesn't differentiate if you have 2,000 or 2 million patients. You know, it's all about the science at the end of the day, and you can make some scientific breakthrough even when you decide to spend the time on such a ultra rare disease like PAX2 syndrome. And at some point, we decided to make things more official and kind of, you know, collaborate even closer with the community. So as I mentioned, we do have a couple of families which we are connected to, but also a couple of scientists which we are connected to. And at some point, we decided to kind of, you know, bring them as much as we could bring them together to Poland. So actually, we had a couple of people flying all the way from the US, from UK, from the Netherlands back in September last year for our first conference, which was mainly focused around the PAX2 syndrome, but we're also talking overall about the rare diseases and what is happening, you know, in both US and Europe and all across the globe around the rare diseases as such. And before I move to the last slide around the advocacy that we are doing in Poland and in Europe, let me invite you for a small video, and hopefully it will be working and the audio will be there.
So welcome on the conference.
I'm just amazed by how many strides have been made in such a short time and how many people have been activated to the cause.
The scientific level was very, very high. I got to interact and meet a lot of very interesting people. I think I come back home with a very renovated vigor, let's say, to work on PAX2 on a lot of rare disease challenges.
That also allows me to see the patients, their problems, their disorder or their disorders in different ways that would allow for different approaches and different treatments.
I'm surprised. I'm surprised because I find that it's extremely incredible how you manage to bring, even though it is an ultra-rare disease, small community, that you manage to bring so diverse people with such a different experience, with such an excellent scientific talks, with people, you know, with really from the diverse horizons.
So I hope the audio and the video is working. And the last thing I wanted to tell you is that, you know, when you focus on the rare disease, when you focus on the ultra-rare disease, very often naturally you start to advocate for the whole community. So obviously 80% of our time we are spending on PAX2, but every month it's moving towards, the percentage is growing towards overall rare diseases. And we are advocating around it in Poland, we are advocating around it in Europe and also in the US. For example, here on the left-hand side, you can read that we were invited to open the Symposium of UK platform of nucleic acid therapies as the main family speaking around the progress we have done. On the right-hand side, on the top, you can see us talking with actually the Polish Minister of Health, Izabela Leszczyna, about the need for the increased public support for the innovative projects on the rare disease therapies. And last but not least, what is for me very proud is, despite the fact, again, we are talking about the disease where we have today 7 patients in Poland and roughly 150 to 200 diagnosed globally. So you could say, hey, we are talking about very small community in Poland, but despite this fact, we were noticed by the Ministry of Health and Polish government. And my wife, she was invited to join the Council for the Rare Disease at the Polish Ministry of Health, which is very prestigious council because there are only 3 patient representation there and around 50 to 20 most important professors which are involved, which are actually day-to-day involved in the rare diseases, together with the representatives of the Polish government, which allows us also to advocate for the overall community and access to innovative therapies and innovative projects, because it's not only about therapies, it's also about allowing a regulatory pathway to be able to treat these patients faster. because in some of the situations, we actually sometimes have the treatment, but if you don't have the regulatory pathway that allows you to move fast, you tend to lose time with the treatment being there on the shelf. So so this is also what we are doing. We are advocating for overall the community, which also is important right now in Poland, because we are leading the European presidency, which also is allowing us to to target and focus on more and more people, not only in Poland but in the whole European Union and Europe as such. And that was it. So thank you for having me here. And again, I'm super happy that I was able to talk once again on the iXCells Day around the rare diseases, because I was able to talk about it 2 years ago. And once again, I want to say that despite the fact that we are miles and miles away from each other, it didn't stop us from collaborating together. It didn't stop us, the parents, from jumping on the plane and flying all the way from Warsaw, for example, to Boston for the NRM meeting every year, or meeting other scientists in a different part of the world. So thank you for that, and I leave it to the last presenter from patient's perspective.
Thank you, Peter. Thank you, Peter. So that's another very, very inspiring journey by the patient. So we've been seeing so many parents that they are switching themselves from a non-biology background into within a very short time frame become a PhD level scientist. So we have been hearing so many stories, and our next speaker is also from a similar journey, but a little bit distinct journey. Noah Auerhan. He is one of the earliest N-of-1 patient parents collaborating with our organization for developing personalized therapy. He is a serious entrepreneur in tech. After his daughter, Ellie, was diagnosed with Rett syndrome, he devoted himself to find treatment for Rett syndrome. He began by identifying repurposed drugs that had improved Ellie's phenotype and quality of life. And over the last 2+ years, he has been developing a series of novel compounds that are working in Ellie's cell lines and are now beginning animal studies. He founded Uncommon Therapeutics, a VC-backed startup, to pursue his effort to build the curative therapies for Rett syndrome. So,
thank you. Thanks, everyone. First, thanks to a lot of people in this room doing actual work for us. So that's really, really exciting. So started a company called Uncommon Therapeutics to basically kind of house all the work that we're doing. As Yanwei mentioned, basically my entire adult life has been building companies in tech and initially e-commerce and then health tech. Started my first company actually down here in Pacific Beach. So was here from 2009 to 2015 roughly. And then sold that company to Rakuten, and then took some time off, started another company in health tech, and tell you about my journey from there. My co-founder, Ryan Lim, is spent about 15 years kind of studying CNS diseases at UCI. And most recently at Module is working on an ALS drug that's going through IND enabling right now. So a bunch of really awesome people behind us and great partners that we've collaborated with over the last almost 4 years now. So my background, I was, while I was building my last company, my daughter Ellie was diagnosed with Rett syndrome in 2021. And so I stepped back from my role running that company to focus all my time on building a care plan for her. When you get diagnosed with something like Rett syndrome, which is more common than a lot of the other diseases that folks are talking about, still the general recommendation is PT, OT, and pray for gene therapy is essentially what a doctor will leave you with. And that just didn't really work for me. So Rett's caused by de novo mutation to MECP2. Basically imagine your child is developing normally, and then around 18 months, they lose the ability to walk, speak, and use their hands. And then there's essentially a cornucopia of terrible things that happen to them: seizures, scoliosis, dysphagia, on and on. it is a neurodevelopmental disease. So in 2007, there was some exciting work that showed that simply replacing the gene could rescue the phenotype in mice. And so that kicked off a lot of funding. And that was part of my kind of experience going into Rett syndrome, which is, one of the first day or so, Googling or going to PubMed and you search MECP2 or Rett on it, and you'll notice 3,400 papers, 200 new ones every month. At the time, no available treatments. So this is like the dream that everyone would want, their rare disease to get this kind of attention. And yet PTOT and pray for gene therapy was essentially all you could ask for. So I think it's a lesson as we figure out where we prioritize our efforts in terms of what are we bringing to patients. Rett does have its first approved drug. We'll talk about that a little bit later now in 2023. But basically, I dove in deep. I probably read about 300 papers in the first 9 months, met with every expert until I became one. And, it seemed obvious to me that we could not develop a gene therapy in a timeline that would be able to slow down this regression, this loss of skills that happens over a period of 3 to 12 months, really. But we could try to bring, drugs off the shelf. And so that's what we did. We developed a thesis around oxidative stress, neuroinflammation, a neurochemical imbalance that exists within Rett syndrome, and a lack of growth factors. And so we attacked that with a handful of repurposed drugs in series, and a couple of those, medications that have been explored in Rett syndrome in those 3,400 papers, really purely based on lit review. And a couple of which, Ellie is the only girl in the world with Rett syndrome currently taking. And so while Ellie is still limited by the disease, here's just a video I took of her the other day walking around her yard. Ellie is 5 years old. She can walk around. She does have an apraxic gait. She has limited hand function, but can touch for yes and no. Limited speech, but things like yes, no, more, done, I do. And you'd be amazed at how helpful those things can be when she can get them out. She doesn't have seizures. She doesn't have dysphagia. She's on her height chart, weight chart, head growth chart. And she's using her eye gaze and is also learning how to read. So her life doesn't quite suck the way that we thought it would, and our life doesn't suck quite the way we thought it did. And that was kind of like the first kind of 18 months was getting to 18 to 24 months was getting to a place of like stability around this. And we're fortunate because it's a neurodevelopmental disease. I think if you're dealing with a neurodegenerative disease, it's a different story in terms of the timelines that you have. But this is giving us an opportunity to then go to the next step of, can we attack the underlying cause of the disease? In this case, the gene of interest, MECP2. So could we leverage, our expertise in the latest technologies to target the underlying cause of the disease? And the more and more time we spent kind of thinking about this from both a scientific perspective on the disease, but also a business perspective on the disease, on how to attack the problem, is essentially, thankfully the investment community has spent billions building lots of new technologies and ways to build drugs. It's really awesome. And so we have, all these new modalities to attack the problem, yet many of them are not reaching our patients. And then also we have, a lot of really great partners who are building capabilities like here at iXCell to do specific activities for us that we don't have to build our own lab to do. And so, can we leverage these technologies, tools, and really great people that are available to us to guide us? And so, what we believe is, an alternative perspective is maybe the platform actually was never the technology, it's actually the disease. And in a disease like Rett, every single drug you would bring to market would be worth billions. And the reality is we know that these rare diseases typically require multiple drugs. That's what we're seeing. And so we think it actually makes sense to build a company there. And that is actually where the value creation point is, and likely better for investors because there's just so much dilution that happens here during the platform creation part. And so we've been proving that out. So for about $1 million, we've developed 3 different approaches to addressing the gene of interest directly and across 3 different modalities that are all working in patient cell lines. So we have an ASO that will increase MECP2. Basically MECP2, the mutation of it, it's a transcriptional factor, kind of epigenetic chromosomal regulator, really important in the brain. And basically, the mutation causes it to lose affinity for the DNA, and then it gets downregulated by 70%. In 2017, a researcher out of UPenn showed that simply increasing levels back to normal was sufficient to rescue the phenotype, because it kind of does this, and that's good enough. And that's a much simpler problem in general than trying to replace the gene everywhere. And when you have different communities, it probably took, I think, the Rett syndrome community like 5 years to adopt that into the orthodoxy, where everyone finally agreed on the research. But meanwhile, that seemed like an approach we could take. So we designed an ASO that would bind to the 3' UTR and increase expression of MECP2. And also we did a large screen looking for basically existing repurposed compounds that could also increase MECP2. And then most recently, got some CRISPR base editor data back, working with the folks here, and in collaboration with some folks formerly from Beam to help us design a base editor. And now we have one that's hitting the mutation. Her mutation is the most common Rett causing mutation, which would account for about 10% of Rett syndrome. And it's correcting about 50% in the cell lines. And so all of these we're working to basically take into the animal models of Rett syndrome. Meanwhile, just because we just started at Y Combinator, we're going through their process and have access to like incredible computational talent in there. And so starting to look at ways, if there's ways to use AI to basically predict compounds that could stabilize the protein as well. So one more project to keep us busy. And so why we're approaching this a little bit differently. So one of the things is, I really don't love the word rare. I think it connotes something that will never happen to you. That's why we call ourselves uncommon, because Rett is not rare. It impacts 1 in 10,000 girls. This is the first Rett approved drug. This is their first full year. They've done 350 million in sales. This is the chart that got them approved. This is the chart on a y-axis that is a full y-axis in terms of the impact on the phenotype. So pretty negligible. Meanwhile, 85% of the patients had diarrhea during the trial. So it's questioned whether it's actually was an unblinded trial. Anyways, that's what's available to us. I think what it does show is they at least know how to really run a trial. And there's a strong desire for treatments in this community and the willingness to put up with shit. So I didn't mean to say that, but no, it's and this is not unique to Rett. This is all of us. And so you have a lot of companies. You have Taysha and Neurogene that are working on gene replacement. It's very exciting for our community. We're seeing those go into phase 1. We're seeing early data off of that are very, very small ends. You're seeing some improved hand function. That's encouraging, but you're not going to transform that full chart. We might see a meaningful step, I think, with the gene replacement, but the reality is AAV gene replacement can't get to enough cells with a single intrathecal or ICV injection. It's just not possible. And so we already know that this disease is going to require multiple more modalities until we get to the finish line. So this is the work we're doing, and brought in really excellent people who have done this kind of work before. And basically, now that we have our 3 candidates, we're taking all of them into animal models. So the ASO is in partnership with Loyola. They're bringing that into mice right now. It's already being injected. And the small molecule study, we're doing our first target engagement study right now with Cyogen. And then we're starting AAV manufacturing on CRISPR, with the goal of doing talks over the following 18 months and then entering phase 1 soon thereafter, and trying to collect some early patient data and then raising the additional capital that we need. Just quickly, some of the key experimental results. This is our most recent base editor data. Really excited about the over 50% editing and the no bystanders, especially in the context of this mutation. There's only 2 non-A's on either side. So the fact that we could hit that was good luck, but also working through a lot of combinations with the folks here at iXCell. We also designed, these are some of the ASO data that showed increasing levels, 1.5x or greater. And then our small molecule work and even seeing some correction of the phenotype and other downstream genes that we did. That's what we're up to. very much.
Thank you, Noah. We are closing our first section. We will have 20 minutes Q&A. So can I invite the three speakers here facing the audience, and also Peter, if you're online. So we're gonna take questions. So there are several questions from online, but I would like to see anybody here in our room that have questions.
Okay, hello. So I have one question for Yue and another for Noah. I'm going to start for the one for Yue. So I was very excited when I saw your phenotype in the microcephaly organoids. I was doing my postdoc at Mootri Lab like a few years ago, and I actually used the same, I actually look at the same phenotype in other disease. So when I saw that violent plots, gave some memories. Did you see also any other kind of phenotype apart of the organoid size? Did you also look at electrophysiology, or did you look at any other stuff?
Yeah, we did look at electrophysiology. Unfortunately, there was a lab-wide incident which killed all the organoids partway through the project. So, that luckily we got the microcephaly phenotype and the rescue study done before that happened. But by that point, it didn't make sense to restart the whole thing. But we were looking for electrophysiology in the thank you, Leo, in the cells as well as the organoid. So it was on our radar.
So with that data, then it was the only kind of data you had in order to push it through the FDA and then to go to the clinic?
That's correct. That was the only efficacy data we submitted to FDA. And in fact, if you look at the N of 1 ASO guidelines, so they only apply to N of 1 ASOs, they do not require efficacy data to approve a phase 1 N of 1 ASO. So that is a very kind of special exception that FDA is making, and I think it's a good decision on their part.
Yeah, indeed. Thank you. And then also for you, Noah. Hi.
Hey, good to see you.
Yeah. I see that at the end that you are looking at 3 different strategies, no, in order to tackle the disease, no, using ASOs, using small molecules, and then using CRISPR in order to deliver to the cells. This is a little bit more speculative. What do you think is going to be the most efficient approach?
Yeah, so I think the highest probability is the existing repurposed small molecule that has the highest probability of getting into patients soonest. But the way that we're thinking about it is not that they're like separate shots on goal, but that they're a series, and we're not done. And I think each drug comes with its own challenge, right? An ASO, it's can we get to enough of the brain regions that matter? An AAV, similar but different brain regions, potentially depending on how you did the injections. BBB small molecule, well, how okay, it's a repurposed drug, so is it just really going to be as efficacious? Do you build a custom drug? It's well, then you're going to do all the other med chem afterwards. So I think it's if you just look at what's happening with SMA, where it's multiple drugs, everybody's kind of working the problem with multiple drugs. So I have what I think will be the first, but I wouldn't say, I think it also that I believe will modify the phenotype sufficiently to get approved. But I think there'll be a series of them in the end.
Okay. Thank you very much. Other questions in the audience?
There are a few questions from online. The first question, I believe, is for Yue. It's a very practical question from Anna Rita Morella. So she said, so can you summarize the timeline and money spent during your Odyssey? I would love to have an overall idea of what to expect.
Yeah, so timeline was we got diagnosed at month 4 when Leo was 4 months old in February of 2022. It took us about 4 months or so to pull all the partnerships and all the researchers together. And from there, it was about 12 to 13 months before we got into the clinic. And we had multiple repurposing efforts going during that time as well, knowing full well that that is the fastest way to some kind of a therapeutic. Unfortunately, most of the repurposing is literature based, right? And so this protein is so little studied that there was basically one paper on TMP02. That was the paper that established it as a disease-causing gene. And so, unlike Noah, I had one paper to read, which was, it's good and bad, right? So after reading that one paper, I knew, I became the expert or one of the experts, right, after reading this one paper. But the other downside was there was very little to go on in terms of finding a repurposed drug. In terms of budget, I'll just say that it was in the millions and not the tens of millions. So it's in the high millions. And again, if you look at the Gantt chart, or there's so many dead ends and there's so many things that did not work, and that, a lot of if you take the critical path, it's in the low millions. But if you wanted to fully kind of make sure that, I wanted to make sure Leo was covered in all these eventualities, it was in the high single digit millions.
Yeah, I have a question for Noah. Yeah, so I know we did some ASO screening and identify a couple of ASOs which can increase the target protein by like 1.5, 1.7 fold. And that's your goal, right? You want to increase at that level, a small change would be enough. But since I have worked in a pharmaceutical company, in general, such a small assay window is very, very challenging for translational science. Yeah. How can we maintain such a small effect from in vitro cell culture system in the human? We see the similar effect. And also, I was wondering whether if we overshoot, let's say we overexpressed by 2-fold or 3-fold higher than expected, is there any adverse effect? So any thoughts on that?
Yeah, so a few things. So one is the ASOs that we designed. What was really interesting was while we were doing that, we found another researcher out of Loyola in Chicago who's doing the same thing. And so he had similar ASOs that were kind of in that 1.5 to 2x range. His appeared to be less toxic than ours. So we're basically in licensing that work right now. And he's right now bringing that into animals. I think it, mechanistically, so we're trying to inhibit a single microRNA. There are multiple regulatory microRNA that are targeting our gene of interest. So I just don't think you can overshoot that much. But this is like some of the other questions that our space in those 3,400 papers, for some reason, hasn't answered. Is the duplication phenotype? So there's a MECP2 duplication disease. But it's unclear whether if you introduce it postnatally that there's even a phenotype, which is like a strange, simple question that we probably should have answered a decade ago. So we don't know. The kind of general thinking is that it doesn't matter postnatally. But there's at least high confidence there's a tolerance zone of like around that 2x number, just staying below that. So I think, again, like mechanistically, it's hard to imagine how you could, just by inhibiting a single microRNA, that you're going to overshoot like 4x. It just doesn't seem possible.
Hi, thank you for the inspirational start to the day. Quick question in regards to, we've all spoke about the importance of setting precedent with the regulators and gaining access to the work and the advancements that we're making. It's a general question to all of you. It's, in your experience being so close, how have you seen that changing over time? Is it getting better as we build better tools, better data? Are we really getting to where we need to get to, which is getting these things into the patients? And I don't know how you want to answer that. It's really a broad topic.
We're a lot further along in the process. We are not close to IND enabling studies quite yet, but I hear from lots of fellow rare disease leaders, and it seems like there's some slow progress. I mean, everybody's doing listening sessions, engaging as early on in the process as possible. And there's still a lot of work to be done, you know, letting regulators know what are meaningful changes to the patient community and the risks that we are willing to take because of what our kids and our families go through. So yeah, I think baby steps, but we have a long way to go.
So I think there is some progress. It's slow. It's almost like one step forward, one step back. Or I mean, 2 steps forward and 2 steps. I don't know. It's like 1.8 steps back. So it's like, yeah. So and I think maybe the most helpful thing I can say is this: physicians have a lot of leverage in these conversations. So for Leo, he had a neurodevelopmental disease, and it puts a physician in a very difficult place because when the patient has a neurodegenerative or a terminal condition, there's a much higher willingness to take risk. And I think that is probably where we will get the most progress is for the physicians, the principal investigators who are treating the terminal cases. I think we can get the most flexibility on precedent there, and then try to push that forward to other patients. In Leo's case, I was kind of, I don't know, like, I would say I didn't regret that he was neurodevelopmental instead of neurodegenerative, but I was wondering, like, if this had been neurodegenerative, would he have a better outcome because we could get the drug to him sooner, and that's just a really weird and convoluted, logical place to be in that the system, the regulatory system and everything has set up for the parents.
Just from my experience, we did expanded access, which is compassionate use for one of the drugs. And it was surprisingly easy. There's like a phone line you can call FDA on and get approval over the phone to start using a repurposed drug. And so I don't know that clinicians realize the power that they have, which is if they think it's in the best interest of the patient and the patient properly consents, it's a phone call to introduce a repurposed drug.
And I think that actually applies to new drugs too. If the physician is willing to put their career and their reputation on the line and say, I'm willing to go out ahead of the FDA here, they can get through these barriers.
So that's what I meant. It's phase 1. If it's been dosed in a single human, it is now eligible for a phone call to FDA.
There are more questions. So I believe the question is for Noah. So this is from Anna again. We have a community that says they really want treatment, but they don't engage as much as we would like to get the data fast. Any tips on how to ignite the community from saying they want treatment to help and do the work to actually reach them? Fundraise, data collection program, etc.
I don't think I've actually done a good job of that. I think Michelle's done a good job of that. I've been among the fortunate to have fallen into a community where a lot of people have already done that legwork and kind of community building, natural history studies. So like I got to skip that part, and I think.
Sorry, can you repeat the question?
So basically, how to encourage the community to gain data faster.
Just outreach, a lot of Facebook messages, a lot of, just community engagement. Again, engaging the scientific advisors and the researchers to communicate directly with the patient communities. That goes a long way. That really increases their engagement and motivation. But it's just, building those relationships, building trust, and taking the time to engage with the community.
I have a question for Piotr. Are you still online?
Yep, I'm still online.
Awesome. Yeah, I have one kind of scientific questions and one non-scientific question. So the first one is, I saw you have done the cell painting assay. My question, I do see you have several candidates, like 23 candidates. So how do you validate those candidates with more physiological relevant assay or any phenotype related assay? Because in general, for these unbiased assays like cell painting, you can always see some difference, but the difficulties or challenges is how to relate those principal component analysis to a particular phenotype.
Okay, so let me start by saying that in our case, what was very beneficial was that we have twin sisters. Despite they are fraternal, but they carry, the DNA from the same parents. And at some point, we decided to approach the issue from the, let's say, precision medicine point of view. So actually, we decided to only do the cell painting on Lena and Zuzia cells, and we were quite struck by the fact how fast we were able to see the phenotype using the machine learning between these two cells. Basically, at the end of the day, comparing morphology of the cells on a very different levels, and it doesn't happen very often with the rare diseases. Sometimes you look at the cells, even like the fibroblasts, and you see almost no difference or no significant difference. In our case, because probably because of the fact that we are talking about the twin sisters, yet fraternal, we were able to quite quickly distinguish the difference on the morphological level. And to your point, at the end of the day, after screening 6,000 plus FDA approved drugs, we are currently looking at the ones that are moving towards, we're looking at the drugs that are moving Lena cells as close as possible to Zuzia cells. And obviously we'll do at the end of the day some filtering regarding toxicity, regarding the side effects connected to that. And we want to filter down the list to 5, 6, which we can use on the mouse. However, if we were approach the whole issue from biased approach, meaning, hey, let's look at the publication, what we know about the PAX2 syndrome, we would never ever list these 23 compounds. They were like super surprising to basically everyone involved. We started to classify them in some mechanisms of actions. So it's not like we're not talking about 23 totally different drugs with 23 totally different mechanisms of actions. Actually, when you start to kind of gather them, you start to see some pattern. But if you would approach it from purely publication approach, so biased approach, you wouldn't pick any of them. So that's kind of the where we are with this. So at the end of the day, answering your question in a very simple way, we are going to focus on the ones that are moving Lena cells as close as possible to Zuzia cells, based on the 3 replicates we did during the final screen of the drugs from the FDA list.
Thanks. So I guess my second question is a little bit associated with the first one, and also a very general question for all of the foundation leaders. So because you guys did a tremendous job to join all these patients globally, but eventually all these studies, it's going to be case by case and focus on one patient, right? So how do you really come up with a strategy to prioritize your pipeline, and how can you scale up or make sure the resources spending in one particular patient can eventually benefit to most of the other families? I think this is I asked the question to several financial leaders, but I know it's very, very difficult and kind of ethical related stuff. So I'd like to hear you guys as a thought leaders.
So my thinking about it was that if we could, get enough efficacy for Leo, then we could prove that these other children deserve a chance too. And then, along the way, if we publish and open source, so to speak, what we did, then that would be the, giving back to the community, and people could take what we learned and use it, themselves. I think, in terms of how it actually played out, we got the drug in Leo much later than I would have wanted. And, the efficacy is, I mean, I'm deeply grateful for any efficacy and for any, you know, avoiding adverse events. But, you know, it might not be so compelling to a payer or insurance. But, you know, there is the open sourcing and we've committed to publishing our IND documents, which is often a very big barrier to, you know, getting into the clinic. And so that is the thinking that, like, you know, it's a learning experience. And if we give the learning to the community, then that will help, you know, every single foundation get to the next step.
So I start with that your fiduciary starts to your child. And so that's kind of how I initially approached the problem. And I made a bunch of effort to try to share what I've done from a repurposing standpoint with the community pretty unsuccessfully. I think that the community, there are folks trying to run trials with some of these different meds that she's on. Some of them, they've already ran great trials in other countries. And, you know, every kid with Rett should be on some of these drugs. And it's just not the standard of care here in the States. And that's a really hard problem to change. It's like a societal thing. I think what I've seen a lot of rare disease parents is they'll spend a bunch of time trying to run a repurposed trial to prove that a drug that their kid's already on and kind of some folks in the community are already on works so that it can become the standard of care prescribed by doctors. Just a lot of effort to do that work. And, you know, one of the things I talked to David Fagenbaum, who's, you know, probably a lot of you guys know, but like Kurt himself of Castleman, is it's like, well, David got to the finish line so he can help everyone get there. And like, we're still running the race. And so it's a complicated dilemma, I think, to the point of prioritization for us. And so definitely could be better at sharing, but I think as we, you know, prioritize, like now I'm, you know, once you start to take on capital, you have to, your fiduciaries change. If you take money from other families, then your fiduciaries change. If you take money from investors, then your fiduciaries change. And so you just have to be, I think, comfortable with what they are.
Thanks. There's another quick questions for Noah. He's asking about a compound you mentioned, a repurposed drug. Do you mind sharing?
The ones we're on?
Yeah.
Yeah. There's 5 different drugs that she's on. So she's on vatiquinone, which we had to go to FDA for. That's the one that's not approved. It's a PTC drug that they're trying in Friedreich ataxia. Dojolvi, which is an ultragenics drug for medium chain triglyceride, so reduces oxidative stress. There's a doc at Emory who ran a 12-patient trial and still hasn't published on it. And at least anecdotally, it seems that it, you know, in his study showed reduced seizures in that patient population. The third is mirtazapine. That's the one that's like blows my mind. That isn't the standard of care. There was an 80-patient study in Italy, which is pretty robust. Showed improved sleeping, reduced apneas, increased appetite, which are like amazing for a drug that you can literally, you know, get at Rite Aid for $1. And then she's on low-dose ketamine intermittently. Again, they tried to run a study in that with Rett syndrome, but it kind of collapsed during COVID. And then the most recent one was Aricept. That was a study done by actually some of former Mootri people at Viant Bio, and they saw it as one of their top hits, kind of normalizing the organoids. They were trying to run that in a study out of Vanderbilt, and that study unfortunately just collapsed from like they lost their financing from the DoD. So that's like, it's, you know, good ideas that, you know, I don't know, I didn't have to invent, they're there. And but you can see the challenges of getting them to patients over and over and over again. And I think, frankly, it requires like insane willpower to eventually get there.
We have 1 minute to take one last question. So we have online questions for Yiwei. It's also one of my curiosity. So how was AI was used in drug design and development, and particularly how I'm gonna, because in a baby lion project, you are trying to use the data and the experience from Leo's case to help to diagnosis and design the drug in a faster way, right?
So I think in Leo's in making Leo's drug, Crayon Bio, using Crayon Bio's model to generate the leads was the main application of AI. There was also, I also used a few of the earlier protein folding tools to try to model the mutant protein. And I think going forward, in the last 3 years, there has just been a tremendous amount of progress in AI, in biology, well, in everything, but also in biology. And so going forward, I think that the answer to that question is very different. AlphaFold 3, AlphaFold 3 came out during the course of Leo's project. It's not perfect, but it can predict interactions between proteins and non-proteins. That the performance in the model is not so good, but again, the next generation will be better, and so on. And AlphaFold 3 has competitors. They have open source competitors. There's all kinds of, Schrodinger is an older company, but there's just a plentiful number of companies who are approaching biology, chemistry, biochemistry, even cell biology. There's an effort at CZI at building an in silico cell model, and that will just change, it will change everything. And so I'm really excited for, where AI is now. Like, there's also the Baker Lab, they can predict protein binding from the sequence alone, the sequence of amino acids alone. And they can get up to 50%, they can get 50% success, at just picomolar, dense. Anyway, and it's incredible, right? So that's where we are already. And so, even a year or two from now, I look forward to the progress.
Thank you.